back to this lecture in pharmacodynamics. And remember, we started on pharmacodynamics. And so today we want to look at something still under pharmacodynamics, and that is none other than what we refer to as receptor families. Remember, we said that most of the drugs basically work through receptors, yeah, and what kind of receptors, yeah, that is basically what we want to see. And I hope you still remember the definition of a receptor. Remember, we said a receptor is a specialized macromolecule, yeah, upon which a drug has to bind so as to bring about an effect, yeah. And we said these receptors can either be on the cell surface membrane, or they can transverse the cell surface membrane, or they can even be intracellular, yeah, so within the cytoplasm or even within the nucleus, yeah. So basically those are receptors. So in this lecture we are going to look at what are the different types of receptor families. Now we have basically 4 types of receptor families, and of course we are going to see what they are and what examples of drugs follow these, or what examples of drugs act through these, yeah. Now we have 4 basic types of receptor families. We have what we call the ligand-gated ion channels. We are going to see what it means. We have what we call the G protein coupled receptors. We have what we call the enzyme linked receptors, and finally we have what we call the intracellular receptors. Now we are going to be talking about one at a time, how it basically works or operates, and then we shall also see examples. We shall also see examples of drugs that work through these different receptor families. Now let's start with what we call or refer to as the ligand-gated ion channels, and we are going to see that these ligand-gated ion channels work through what we call a ligand. So there is a channel, an ion channel, and once this drug binds to this ion channel, then this ion channel opens to allow flow of ions, yeah, and these ions might be sodium ions or they might be chloride ions, as we are going to see examples of these drugs. Now once it opens, once the ligand binds, and in this particular context we are talking about the drug, in this context is our ligand. Now once it binds, as you can see, it binds and then it opens up that channel, what we refer to as the ion channel. So once it opens it up, then ions flow inside the cell, yeah, intracellularly, and then from there we have changes. Like for example, if it has to do with muscular contraction, then we have a muscular contraction. If it has to do with release of certain maybe certain enzymes or even hormones, then we shall have release of these. And when the ligand has stopped its function, remember we said it does not work forever, yeah. A time comes when it has to go off the receptor, yeah. Remember we talked about bonds, if you still remember very well, and we talked about the electrostatic bonds, yeah, and the hydrophobic bonds. These are reversible. The drug or the ligand does not bind on the receptor forever, and so it has to, it comes off. Once it comes off, then the processes basically stop. In other words, once the ligand comes off, then the ion channel closes. And what are the examples of drugs that follow this? We have examples like local anesthetics, which you know what local anesthetics are used for. They basically block pain, yeah, so they block nervous transmission of pain. And examples like lidocaine, yeah, these block sodium conductance through what we call voltage gated sodium channels in the neurons, and therefore prevent transmission or the interpretation of pain. We also have what we call benzodiazepines. You have heard about a drug called diazepam. These basically work through what we call the GABA receptors, or what we refer to as the gamma aminobutyric acid. And here, these receptors, once the benzodiazepine binds, as we said earlier on, then we shall have the ion channel open, and in this particular context, what flows inside are the chloride ions, yeah, are the chloride ions. So once they flow inside, they make the cell very electronegative and so, for that matter, very hard to excite. That is why patients who have convulsions, yeah, are given benzodiazepine for this reason. And so this is how benzodiazepine is able to work. We also have other drugs or other neurotransmitters like acetylcholine. We shall talk about autonomic, the autonomic nervous system later on. But one of the neurotransmitters, acetylcholine, works through this, yeah, via what we refer to as the nicotinic channels at like, for example, the neuromuscular junctions, yeah. So acetylcholine via the nicotinic receptors also works through this, this receptor, which we call the ligand-gated ion channels. The next that we are going to talk about are what we call the G-protein coupled receptors. Now, these G-protein coupled receptors are also called 7-pass. They are also called 7-pass receptors, or they are also called serpentine receptors. We are going to see why they are called this, yeah. So G-protein coupled receptors, they are actually the most common types of receptor families through which most drugs work, as we are going to see examples. Now, a G-protein coupled receptor, remember we said a receptor basically has two sides, yeah, and a G-protein coupled receptor brings it out very well, yeah, as you can see. Assuming this is our cell membrane, we have what we call and we have what we call a G-protein coupled receptor. So it has what we call the receptor site, yeah. It has what we call the receptor site, and then it traverses the cell membrane 7 times, yeah. 7 times. That's why it's called 7-pass. Yeah. So 1, 2, 3, 4, 5, 6, 7. Yeah. So it traverses the cell membrane 7 times. That's why we call it a 7-pass receptor. And for the same reason, we actually call it serpentine, because as you can see, it resembles a snake, or the serpent for that matter. So this G-protein coupled receptor has 2 sites. It has what we call the receptor site, this one. It has the receptor site. And this site is where the ligand binds, yeah, or what we refer to as the ligand binding site, yeah. The ligand binds here. And then it has what we refer to as the effector site. Now with the G-protein coupled receptor, we have what we call a G-protein, yeah, and that G-protein comprises of 3 subunits of proteins, what we refer to as it has what we call the alpha subunit. It has what we call the beta subunit. And it also has what we call the gamma subunit. Now the whole of this is what we refer to as the G protein. Yeah? The whole of this is what we call the G protein, which has 3 subunits. It has the alpha, the beta, and the gamma subunit. So this is the receptor site or the ligand binding site, and then this is referred to as the effector. So in other words, effector site. So in other words, once a ligand binds here, as we are going to see, then we see changes within this G protein. So that is why we refer to as the effector site. Now, this G protein is basically, as we said, it's a polypeptide chain, yeah, that traverses the cell membrane 7 times. That's why we refer to it as 7-pass receptor. But also it moves like a snake within the cell membrane, and that's why we refer to it as a serpentine receptor. So it has what we call the ligand binding region, as we can see here, and then it also has what we call the effector site, which is comprised of the G protein. And that G protein has 3 subunits: the alpha, the beta, and the gamma. Now, how does the G protein receptor work? Now, assuming we have our ligand bind, yeah, like for example drug, you have our ligand bind here. When we have our ligand bind here, once we have our ligand bind there, then we have what we call a series of signal transductions that happen. Yeah, what we call a series of signal transduction. A series of signal transduction that happens that eventually reach the effector site, yeah? And once it reaches the effector site, then this G protein receptor will have to dissociate, yeah? It dissociates, and in this particular context, the alpha subunit gives up a GDP to attain what we call a GTP, yeah? So it gives up a GDP to get what we call a GTP. And once it gets a GTP, or guanosine, triphosphate versus guanosine diphosphate. Once it gets a GTP, then it is said to be phosphorylated, or it's said to be activated. And therefore, once it is activated, what actually happens is that we have it separates from the two, yeah, it separates from the 2 proteins. So remember we say this is the alpha, and then this is the gamma, and then this is the beta. So once it is activated, once it gets a GTP, yeah, then it dissociates from the unit, from the whole unit, and then it basically goes to effect changes, yeah? The alpha subunit goes within the cytoplasm to effect changes. So in other words, this one goes to effect several changes or processes. And we are going to see that once this subunit goes, it can go to activate what we call adenyl cyclase enzyme, or it can go and activate what we call phospholipase C, which is an enzyme, located within the cell membrane. Yeah, and as a result, then you have several changes, as we shall see. Like for example, if we look at one, where it activates phospholipase C. Once it leaves, it goes and will activate phospholipase C. And once phospholipase C is activated, this phospholipase C basically goes and cleaves, it cleaves part of the cell membrane, and once it cleaves that part of the cell membrane, it is interested in a substance or a phospholipid referred to as PIP2, which we also call phosphatidylinositol 4,5 bisphosphate. And so once it cleaves it, you have two substances that basically come out of that. So you have what we call DAG, or what we refer to as diacyl glycerol, and then we also have what we call inositol triphosphate, or referred to as IP3. Now these are referred to as, these two are referred to as second messengers. Second messengers. In a minute we are going to see what it means, or what we mean by a second messenger. So these are referred to as second messengers. Now it is these particular ones, these particular second messengers, that will bring about several effects. Like for example, once IP3 is released, it basically triggers release. It causes opening of the calcium channels, so that we have massive release or massive entry of calcium within the cell. And of course, once calcium enters the cell, depending on what that cell is meant to do, then you have the other or the subsequent changes that basically happen. And then diacyl glycerol, for it goes an extra mile of activating another enzyme called protein kinase C. And once it activates that enzyme, protein kinase C, that enzyme is active to cause or to bring about further changes. Like for example, if there is need for protein synthesis, if there is need for muscular contraction, if there is need for, and depending on what that cell is meant to do, or depending on what that drug was meant to do. So these are referred to as the second messengers, and in a minute we are going to see what a second messenger is. All right, so of course we said that the ligand does not bind on the receptor forever. Remember, we said these are reversible changes. And so once the ligand separates from the receptor, then what actually happens, the processes go back. The alpha subunit that was being activated by the GTP, it gives up the GTP now for a GDP, and then it goes back into that trimer, or that complex of 3. In other words, it attaches back to its original position or original location, waiting for yet another drug, or waiting for yet another ligand to bind. So basically that is how G protein coupled receptors work. And if they do not activate protein lipase C, then we also see that this alpha subunit can go ahead and activate what we refer to as adenyl cyclase. And of course we shall see this more. And once adenyl cyclase is activated, then we see conversion of ATP to what we refer to as cyclic AMP. And this cyclic AMP actually plays a lot of functions, or a lot of roles, as far as that cell, that primary cell is concerned. So that is how G protein-coupled receptors work. Remember, we also talked about ligand-gated ion channels, and now we have talked about G protein-coupled receptors. When we come back, we shall talk about other types of receptors, but also what we refer to as second messengers. Thank you so much.